Thank you for coming this morning. We are pleased here to have our guest, Professor Sean Edward Lawler. Pronounced it very well. That's okay. Yeah. So basically, I know Sean as a person more than a scientist in the beginning. We are really pleased to have him here. So Sean, he's an associate professor here of pathology and laboratory medicine at Brown University. We know each other actually from Boston, from Brigham Women's Hospital, where we had a lot of interesting discussions about philosophy and science. And he's a co-leader of the brain cancer research program at the Ligoretta Cancer Center. He earned his PhD in crystallography,
in the crystallography department.
In University of London in the Birkbeck College, and has previously held academic positions in Harvard, of course, where we know each other. Also the Ohio State University and the University of Leeds. You are originally from Leeds.
I'm originally from Leeds.
With American accent. Actually, his research focused on glioblastoma biology, tumor immunology, and novel therapeutic delivery across the blood-brain barrier. So Dr. Lawler has authored over 130 peer-reviewed publications with H-index over 61 and lead multiple NIH funded translational neuro-oncology projects. He also served on several international scientific advisory boards and editorial panel in neuro-oncology. Thank you very much. We're really pleased to have you here. Thank you.
Yeah, thank you, Nabil, for that kind introduction, and also for the invitation to come to Sevilla, where I've never been to the south of Spain before, and I've enjoyed every moment since arriving. I love the weather you have. But anyway, it's very familiar weather to me, in fact, because I'm from the north of England originally, where it rains all the time, and blue sky is extremely unfamiliar. And so you'll know that the English do love Spain. I'm not sure how much you love the English in Spain, but we do love it here, and I'm very thankful for the opportunity to come and really, try and build some bridges between our groups. Nabil is obviously working on brain cancer, which I work on for the last 20-something years, and so I think we have some really great opportunities to connect and work together and make our projects more powerful. And the point of all of this is really to improve the outcomes for patients with these diseases. So glioblastoma is the most common of the brain tumors. I think in the United States it's 12,000 cases per year, and all of those patients practically will succumb to the disease within 5 years. There's a 5% survival rate after 5 years, a median survival of only 15 months, and that has not changed. Even though we know all the genomics of these tumors down to great detail now, we know about the tumor microenvironment. There have been hundreds and hundreds of clinical trials, and nothing has had a major impact on the disease. So we have a major challenge between us by the time we retire, which is a few years away, but we need to have an impact on this disease. So this is what it's all about. Right, so this is actually a picture of glioblastoma illustrated here. You can see this is an autopsy sample of the human brain with a very bloody large tumor in it. And one of the really key features of this tumor is that it spreads around the brain. It has this natural propensity to the cells will migrate and form colonies elsewhere, so it makes it surgically impossible to remove. So this is a disease that requires some other intervention in addition to surgery. So my lab really focuses, I'll go relatively quickly. I want to get through at least 2 talks in the next 45 minutes. So I'll try not to make it too much, but our goal is to we're looking for mechanistic insights that will guide new therapies for glioblastoma. I'll talk about drug delivery. Getting drugs into the brain is a challenge. I'll talk about a drug we work on which is called indirubin, which derives from a Chinese medicine. I won't talk too much about this question, but this is an important question. Starting the fire. Can we heat up these tumors? Can we make them sensitive to immunotherapies, which have made some great impacts in some other tumor types, but have failed to make an impact in this family of tumors? And I'll talk about cytomegalovirus, which we have discovered may have an important connection with glioblastoma and other tumors. It's a very common virus, and we'll cover that in the last half of the talk. And my lab is now based, I used to work in Boston. This is where I met Nabil, and if you come straight across, I guess you will reach Barcelona eventually. And Providence is about 50 miles south of Boston, on the way to New York City. It's a smaller city. It's got about 200,000 people, and it's really one of the major things in Providence, Rhode Island, the smallest state in the United States. One of the most important things is the university called Brown University. It's one of the oldest universities in the United States. It's classified as an Ivy League university. It prides itself on education of undergraduates, really, but it does have a growing biomedical center and lots of local hospitals and patients. And I'm very excited to be part of the growth at Brown. Just one slide to cover glioblastoma, really. So here we have some normal brain on the left, and glioblastoma belongs to a family of tumors. If a patient presents with glioblastoma, which is classified as a grade 4 tumor, there's no record of a lower grade tumor. It comes as glioblastoma, and it's diagnosed as glioblastoma. And the mutations typically seen in that are in the TER promoter, which is about 90% of glioblastomas have a mutation there, which drives immortalization. Lots of tumor suppressor genes like p53 and p10 that we understand, and turning on of receptor tyrosine kinases. So like many cancers, these pathways are altered. The other family members take a little bit longer to develop. So oligodendroglioma and diffuse astrocytoma both have a very common mutation in a gene called isocitrate dehydrogenase, which defines these tumors as being distinct from glioblastoma. And these can take a long time to evolve, a decade or so, but they eventually will advance to a high-grade tumor and have the same consequence, which is tumor recurrence of a resistant tumor that does not respond to therapy and leads to the demise of the patient in probably a really horrible way as well. So our goal is to try and find ways to stop this happening, or at least extend patient survival significantly beyond what it is now. And so the standard of care people receive with glioblastoma, so you present with the disease, you might get an MRI scan. The doctors don't really know it's glioblastoma until they can do the pathology, but you can kind of recognize it on MRI. Anyway, the patients will get a certain maximal surgical resection without damaging patient function. It will never remove the whole tumor because it's invading the brain, and you cannot take out too much of the brain, obviously. And then the patient will get a 6-week dose of radiation therapy plus an alkylating chemotherapy called temozolomide. Temozolomide can get across the blood-brain barrier, and it does provide benefit in some patients, but not all patients. And then over the many weeks after the initial 6 weeks, temozolomide is given every few weeks, but eventually the tumor will recur, and then there's more surgery. Maybe there's a clinical trial, maybe there's some other interventions, but the median survival remains about 15 months. This is the original clinical trial which established this standard of care, which was published in 2005. So we haven't really seen much improvement on this since 2005, except for things like refinements. We can do the same therapy but better, but we can't make a massive improvement yet. And so other things to think about is the patients are all on dexamethasone. Dexamethasone is also immunosuppressive but helps relieve some of the symptoms of glioblastoma. This can affect things like immunotherapy, obviously. And patients will also self-administer drugs. So they'll read in the literature that CMV might be important in glioblastoma. So people will start taking valganciclovir, which is an antiviral drug. And so there's all sorts of complications and other factors to bear in mind. And so just some challenges. So I try to design my projects based on the challenges. So let's define a challenge. One of the challenges is surgical. So the first projects I did, which I'm not really going to talk about today, the first projects I did in glioblastoma were looking at invasion. So can we find things that would block invasion? These were published in 2008, 2011. And then more recently we got into this idea of drug delivery because we were thinking about treatments, but what is the point of developing a treatment if you can't get it into the place? And also these tumors, the biology and the rapid growth, they're very heterogeneous. So each patient has a slightly different version of the tumor. So therapy should probably be personalized as well based on tumor genetics and the location in the brain. There's all sorts of variability. These tumors are highly immunosuppressive, as I mentioned, and also we do need to understand much more. And I think a lot of our understanding moving forward will come from patients, and people like me who are scientists will learn from patients rather than learning from cells. So we'll be inspired by the patients. So I'm going to talk first about the blood-brain barrier, right? So this is a very famous old experiment. done by a famous German scientist. Although it's more complicated, there were many people involved, and this is a great review. It describes all of the different people who were involved. It's like a historical piece. It's quite nice to read. But what the experiment was they injected a trypan blue dye into mice, or rats actually, and they found that it stained everything in the body except the brain. And Paul Ehrlich thought that it was because the dye doesn't bind brain tissue. So another person did an experiment where they injected the dye into the brain, and they showed that it actually does stain the brain when it's injected into the immune system. So this was the sort of first hint that there was a barrier between the brain and the rest of the body. And then obviously many more things happened since 1900 or 1890, and the cellular composition of the blood-brain barrier is now very well defined, and it's basically endothelial cells in the brain have properties which allow them to form extremely tight junctions between them, which stops anything pretty much getting through that barrier. This barrier is enhanced further by interactions with astrocytes which surround the thing, extracellular matrix. There's also a matrix inside the lumen of the vasculature called the glycocalyx. So there's a lot of layers of the barrier which really stop cancer drugs getting in, stop toxins getting in, but also will allow nutrients and the right things to get in and kick out toxins. And this is just some usually I show cartoons, but recently I've been showing these electron micrographs because I think they're more informative than a cartoon shows a straight line between the endothelial cells here, whereas actually the tight junction is a bit more complicated than that. It's more of a circuitous kind of structure with tight junction proteins towards the lumen, and then proteins which are slightly different called adherens junction proteins further inside. So there's a complex organization with different proteins organized very, very carefully to prevent things getting through. But also in the case of, say, if an immune cell needs to get in or something else, then this can actually be opened by various regulatory processes to allow, say, a monocyte to get in if there's an infection or something like that. So it is regulatable. This is it's too much already, this one. But this is I put this in because it's the most beautiful diagram of the blood-brain barrier that I've found in the literature. It's from the lab of someone called Chenghua Gu, who's at Dana-Farber Cancer Institute. These really are the leading lab, in my opinion, on blood-brain barrier. It really just shows that structure. These are tight junctions towards the lumen. The lumen is here, the blood is here, flowing along, and the blood-brain barrier will selectively take in some things by transcytosis. So, for example, transferrin will carry iron through transcytosis, and there'll be a pinching of the vesicle and a transfer to the other side where the cargo is released. And then we have the organization of these different tight junctions. And another important feature when you're thinking about cancer therapy is things like efflux transporters. So even if a drug gets into an endothelial cell, there are transporters which will push it right back out again or modify it in different ways. So here we have a very complicated barrier that we have to overcome, perhaps. There are other ways, but I think we need to think about this barrier for treating glioblastoma. This is a cartoon a colleague of mine made when I said, can you do me a cartoon of the blood-brain barrier? Anyway, it's a wall, right? So the great majority of systemically administered cancer drugs cannot enter the brain, so we have a challenge. Brain tumors, though, they show a variable permeability. So this is an MRI scan of a glioblastoma. And in the center, you can see this enhancing region. This is gadolinium, which is being given systemically to the patient. And this shows that this gadolinium can get into the tumor. So there are areas of the tumor which are a little bit permeable. But this is another form of MRI called FLAIR, T2 FLAIR, which doesn't rely on permeability, but it shows that the disturbance in the brain is much more extensive than you can see with gadolinium alone. And there are tumor cells all scattered throughout this region that are protected by the blood-brain barrier and probably are not going to get removed by surgery either. So these are the cells that I think we need to be getting with drugs that will cross the blood-brain barrier in some way. There have been many studies showing that tumors in the brain do not take drugs up at all to a level comparable with systemic extracranial tumors, which will. So there's 10% less, or 90% less, I should say, compared with other solid tumors. So we have a problem here. There are many ways to think about getting drugs across the blood-brain barrier. So you can actually think about bypassing it completely. You can inject drugs into the spinal fluid. You can use oncolytic viruses during surgery, things like this. See, when surgery happens, there is an opportunity that is not exploited enough, in my opinion. You can add drugs, you can put things in hydrogels. There are many opportunities there. We're going to talk about systemic drug delivery, though, for this part of the talk. If you've got some small molecules that are small or hydrophobic can cross the blood-brain barrier naturally, right? So those ones would be the first choice in a way. But there are many drugs which could be effective that don't have those properties. So how do you deal with that? I'm going to talk a little bit about some BBB penetrant drug conjugates, that my lab has made. There's also the rise of ultrasound. Focused ultrasound is becoming important in the clinic, so to open the blood-brain barrier very gently and safely, transiently, to allow drugs to get in. We're going to talk about something we recently discovered where we think we can manipulate the blood-tumor barrier, not the blood-brain barrier, but where the tumor interacts with the endothelial cells. We think we can manipulate that with small molecules. And then there are various nanoparticle approaches as well. So I'll tell you about one. I'll start off with one project which started about 2016, I would say. And we met Brad Pentelut, who was at MIT. My lab was in Boston at the time. Brad this is the fastest peptide synthesizer in the world. So he's a very well-known peptide chemist. He makes short sequences and does lots of fancy chemistry. And what he'd found was that a peptide called TP10, which is it's a peptide. It's actually it's a fusion of two different natural peptides, but it has a natural ability to cross into plasma membranes, into cells, and through cells to the other side via some helical structure. But they found that if they put these modifications called perfluoroaryl staples, so these are aromatic rings with fluorine in them. If they put those onto the peptide, they can penetrate cells much better. I'll show you an example of that. And the idea of this collaboration was to make peptides that could target. So we actually have some targeting peptides. I won't talk about those because they're not very good. But we're trying to make targeted peptides that will bind specifically to tumor cells and cross the blood-brain barrier simultaneously and carry a drug in. So I'll show you what we've done so far. We actually developed a model of the blood-brain barrier too, which I should tell you about. We call it BBB spheroids. This again was published originally in 2017. And when we did this experiment, I thought, well, we get a nice paper, but the model is probably useless, right? It's not gonna carry on. It's just gonna fade away, and we're going to think about something else. But actually, we're doing this model every week in the lab, and the way the model works is that we take human brain endothelial cells, we take human brain pericytes, and we take human brain astrocytes, which are commercially available from companies. And if you mix them together, 1,000 cells of each type in a low attachment 96-well plate, they'll form a sphere spontaneously overnight. And that sphere has an organization where the endothelial cells coat the outside and the astrocytes and the pericytes are on the inside. So it's like an inverted blood vessel in a way. And what we did some studies here that we published in Nature Communications that show that there's tight junctions, that show that there's efflux pumps, and shows that these things actually do have some features of the BBB. I will not claim it's the BBB. I think that's probably impossible to replicate fully in a simple in vitro system. But it has enough of the features of the BBB, I think, to make it useful, and it's very simple and very cheap to use. So here's the example of what we've done with it. So this is you making the peptides with Brad's lab. So we made several different versions of this penetrating peptide with these perfluoroaryl groups, and we tested them in the BBB spheroid model. And we ultimately these are conjugated to a Cy5 fluorophore, and we saw that this one M13, which is there it's basically has two cysteines here which link the group to the perfluoroaryl group, and this one seems to work the best. When we compared this is a brain with the fluorophore, so we injected a mouse via the tail vein with this fluorescent peptide, and we could see that it was uptaken into the brain quite nicely, particularly compared with a linear peptide of the same composition but without the chemical modification. So we actually went forward with that. This is going through a lot of work in a very short space of time. We went forward with that, and we designed it. This is the M13 peptide that I just showed you in the BBB spheroids, and we conjugated platinum-4 to it, right? So platinum-4 is a prodrug of cisplatin. Cisplatin is obviously used for cancer widely. It's not my drug of choice for treating glioblastoma, but it's a very good drug for doing a prototype experiment like this, right? So platinum is really easy to detect in the brain using mass spectrometry, and it's cheap. So ICP-MS we use to measure the concentrations of platinum in the brain. So this has been a very useful tool to understand how to get things into the brain. So we made this construct, and the cisplatin is released when the peptide gets inside the cell, and you enter the reducing environment of the cell. Chemically, this is released, it is. And so what we were able to show is these are in vitro experiments using neurospheres, and that the neurospheres were getting smaller with dose of the PT4M13 drug. We did some tissue biodistribution. You see, it doesn't get into the different tissues accumulate the drug at different levels, but the only ones with significant changes are really the brain and the tumor in the brain. So this is a magnification of that, showing an almost, I'd say, 20-fold increase of platinum in the brain using the PT4M13 compared with cisplatin alone, and similarly in the tumors. And when we treated mice with intracranial glioblastomas with this compound, we were able to show increased DNA damage. This is a histone H2AX, which measures DNA damage in the nucleus, and we were able to get an increase in survival in animals. So this is where we're at with this project right now. We're trying to get more funding to play around with the system more and build on this prototype. But when we were doing this project, I started to think about endothelial cells, which I'd never really worked on before, right? So, and now all my projects seem to be focused on blood vessels in the brain. It's quite a change over the last few years. And so what we tried to do here, I tried to understand the molecular biology of the vasculature in the brain. And we used publicly available data from the Cancer Genome Atlas, which you could just, in fact, we didn't even download anything. This was just using interfaces. You could type in gene names and pull out lists. And we found that we use these as probe genes, and we looked for transcripts that were associated with these three genes, which are known to be highly and specifically expressed in endothelial cells. And we found a common list of genes here which are overexpressed in glioblastoma and associated with these three. And these, when we put these into a gene ontology engine called String, which is really a nice tool, it's free, we found that they all cluster together, so they have related functions. And the central molecule in this was called cadherin-5, or VE cadherin, vascular endothelial cadherin, which is one of those junction proteins I was telling you about at the beginning. So it's an adherens junction, and it sticks together like that. The same protein is expressed by neighboring cells, and they basically homodimerize it with the extracellular domains to make a junction, which helps hold. And this just shows you about CDH5 quickly. This is data from another free atlas, the IVGBM atlas, and it shows that this CDH5 is highly expressed in blood vessels and vasculature. We did some playing with spatial 10x spatial biology. This shows CDH5 in some tumors from a data set that's available online. And these spots where CDH5 is elevated in these tumors actually correlate with a bunch of endothelial markers as well. Again validating this idea that CDH5 is perhaps important at the blood-tumor barrier. And here is this is also free data. I'm from Yorkshire. We don't like spending money, so we do everything. We find everything free is like a victory. You know? Even though my lab does have money, but free is good. This is the human protein atlas. And this shows CDH5 staining. And for those of you who've seen tumors before, you will recognize that these tube structures are actually blood vessels within the tumor, and they're highly stained for CDH5. Again, it's confirming that this protein is doing what we think it is, that we've identified. Now we're going to take we're going to forget about CDH5. We're going to come back to CDH5 in a couple of moments, but I want to tell you about something a little bit different, indirubin. So we've been working on this I think we started working on it in 2008 because it's anti-migratory, and this was one of the early projects. And we found that this drug called Bayer, which is a derivative of, I'll tell you a bit more about. Indirubin is there. It functions in various different ways, but it inhibits a bunch of different kinases, and these are some of the kinases it inhibits, and it affects a lot of different aspects of tumor biology if you do some assays, and it also extends survival in some mouse models. And we published on this first in 2011. And we kind of stopped because this drug's really insoluble and is very hard to work with, and we didn't think that we would get any further with it. However, this is the introduction to India rubin as well. So it's actually a natural product from the indigo plant and other organisms, marine organisms too. It's found, and it's blue. India rubin is a component of the dye indigo. Indigo is blue, and indigo stains. If you're wearing jeans, indigo is in your jeans. The genes on your legs, not the genes in your nucleus. Anyway, this India rubin is part of the indigo dye, and is also part of a complex Chinese medicine here, which was known to be anti-inflammatory, and so it's been used for thousands of years. Used as a textile dye, economically very important. And then in the 1990s, when molecular biology was becoming easier, I guess, it was found to be an inhibitor of a bunch of protein kinases, including cell cycle dependent kinases, GSK3, and Janus-associated kinases too. So Janus kinases, so they're downstream of things like interferon signaling. It's been used for treating psoriasis as a topical application, and it also has been clinically trialed in China for chronic myelocytic leukemia with mixed results, but there are better drugs, so it really hasn't been pursued for that. So we gave, like I said, we gave up on this drug, but then a guy from a company contacted me and said, we have a nanoparticle formulation that we think will work and allow you to deliver it. So we tested it, and we gave this nanoparticle formulation, which is a bunch of poly PEG and all of these things combined together with the microcrystals of the drug. I'm not a chemist, but this is basically what we did. We took that, we injected it via the tail vein. It was safe. Previously, we'd been doing IP injections of and with a bit of DMSO, not clinically translatable. But here is a tumor. This tumor has a reporter gene in it that measures inhibition of GSK3 and releases luciferase when you inhibit that enzyme. And so when we give a tail vein injection of our drug, we can actually see the tumors in the brain light up because we're hitting the pathway, which I think was a lovely experiment. We did some mass spectrometry, and we were able to show that after 1, 5, and 24 hours, this drug is detectable in the brain and the tumor, and that there's a favorable ratio of brain to plasma of the drug over time. So it seems like it's actually getting into the brain. And it will, and mixed results in these 2 glioblastoma models, which are immunocompetent, which we could see extension of survival in GL261 and a tiny extension of survival in another model, CT2A. In this paper, we look at a lot of immunological readouts, but I'm not going to talk about those because what we also found with, oh, we've got a jam. Wait a minute. Breathe, it's me. Right. Hi, there we go. So what we found many years before we really started thinking about the blood-brain barrier, we did an MRI study. This is a tumor in the mouse brain, and we gave the mouse this Bayer drug. And what we saw was a real change in this. It's called dynamic contrast enhanced MRI, and it's an MRI technique that measures fluid flow in the tissue that you're looking at. And the more red the signal, the higher the flow in this case. And so this is the pre-treatment tumor on the bottom, and this is the post-treatment. I think there's 2 things to note. One is that normal brain doesn't change, and one is that within the tumor we get a very increased red signal. And at the time I thought, this is interesting. And then I went and did lots of other things instead of thinking about this. But then later on, we got a system in the lab called called an ESIS system, which measures transendothelial resistance. So you grow your endothelial cells as a monolayer, and there's electrical sensors on the tissue culture plate. And those electrical sensors will measure the resistance of the monolayer, and that resistance is dependent on the formation of tight junctions. So I thought, well, let's put Bayer on that endothelial. So here we get the monolayer is forming, and you get a plateau. And when we add Bayer, and we've done this many times, this falls off. The cells are they're not dying or anything. We've done all those kind of controls, but the barrier really breaks down, which is great. And so we did, these are BBB spheroids that we used, and we were able to show that, for example, CDH5 staining goes down with Bayer over time. We were able to show that dextran, so dextran doesn't usually get into these spheroids very well, but with increasing dose of this Bayer drug, we were able to see that these spheroids were allowing the dextran in. And there's no cell death involved in these things. So we think this biodrug, in addition to some of the other properties that I've told you about, actually can play with the barrier. So what we learned about, this is more a bit of molecular biology. Yeah, we have some ideas, but we don't really know yet. We know that our drug can inhibit protein kinases. Here are some of those. We know that those protein kinases can drive various transcription factors. And we're diving into this a little bit right now, and those transcription factors can affect the BBB program. But what I'm more interested right now is how do we get this into a clinical situation? Mechanism is important, and we're looking, but what can we do to translate it? And so we haven't translated it yet, but we're working towards that, right? And so we did some gene expression analysis. These are endothelial cells that form the monolayer, that are treated with the bio. We see lots of genes change. So exciting to see that nearly all of our signature of the blood tumor barrier genes go down, which really is relevant, including cadherin-5. And this is cadherin-5 staining in the monolayer, and you can see that it's the Western blot actually is a little more impressive, but the barriers are breaking somewhat after biotreatment. And this is an in vivo experiment, so the green is a GFP expressing tumor. The holes in the tumor are the blood vessels, and we stain them with CDH5 and CD31. You can see the red and the blue completely overlapping. 24 hours after treatment with Bayer, this is the PPRX drug actually, I should say, the nanoparticle formulation. We see that the blood vessels are still there, but the CDH5 staining is gone. So what we were interested in doing was to understand, can we get drugs into that tumor? And the drug we chose actually was cisplatin, because we were working on that already. We have the assay systems ready, but we were interested in other things for sure. And so what we did, this was published this year in Science Advances. There you go, I put the reference up there. This is the nanoparticle formulation kind of in cartoon form without enough information really, but when we put tumors into the mouse brain, and then we did a dose response, so we took 1 mg, 5 mg, 10 mg, 20 mg per kg of the biodrug as PPRX, as this formulation. And what we could see was in the blue here, there's not really a change in cisplatin uptake. So we would give the Bayer on day 1 and give the cisplatin on day 2, and then 5 hours later we'd harvest the tumors, right? So that was the assay setup. And we could see this dose-dependent increase of cisplatin levels, but only in the tumors. So this is very exciting, and we did a survival study based on that, and we get an extension of survival in several models, but this is one example. And so this tells us that we can get non-penetrant molecules into brain tumors using another small molecule that targets the vasculature. And so what we're doing in the lab right now is we're working on methods to translate this. So we're looking at other kinase inhibitors. Kinase inhibitors are already FDA approved. We're screening those. We found potentially some candidates that work similarly to Bayer. Mechanisms are a bit different, but will allow drugs and particularly antibodies into the brain. So this is what our focus is right now, is to try and get therapeutic antibodies into the brain tumor microenvironment, like an immunotherapy or like whatever we want, like a HER2 targeting antibody drug conjugate, something like that. And so that's our main. We're trying to get money for this right now. So if anyone wants to give me about $250,000 would be okay. We will carry on. So that's really the end of my drug delivery section. We'll go on to CMV in a second. But I just wanted to kind of tell you the story of how I got into glioblastoma. So I'm a molecular biologist. I grew up as a scientist in the 1990s. I see probably some other people in the room may have done science in the 1990s, and we used to do things like clone genes because there was no human genome project, there was no databases. It was more like, well, we know something is there, so what is it? Let's find. And it was a lot of fun. It was very rewarding actually, in different ways, right? So I cloned some genes. I cloned one TGF-beta receptor. I cloned a JNK, MKK7, so that's my claim to fame, really. But I was kind of, the Human Genome Atlas project was done, and science changed like that. And I was a bit like, what am I going to do? And so I was working at Mass General, and I wasn't in this guy's lab, but he was in the lab next to mine, and he came in the lab in his scrubs. And I am a PhD. I didn't really know many MDs, and it's very important for PhDs, any in the room, to know MDs nowadays, to learn what they know and what their challenges are, right? And so he came in there. I said, "What were you doing?" He goes, "Oh, I just took a tumor out of a patient." He's a neurosurgeon. It's very, took a tumor out. And then I said, "Will the patient live?" He goes, "Well, he's doing okay now, but in 1 year, he's going to die." And because I'd cloned all these genes and understood pathways, it was that light bulb moment. It took me 1 second. I was like, wait a minute. There's an opportunity here for me with what I know to apply my knowledge. And so we haven't yet had any true success, but this was the moment of inspiration. So I joined his lab and started working on glioblastoma. And I worked with him for 15 years or something. We published 60 papers, which is good, and he does oncolytic viruses and is making great progress there. I decided ultimately to go my own way at Brown, and so. But anyway, he's a very important man in my career trajectory. And while I was in his lab, we decided to address this question, which is the role of cytomegalovirus in the interaction. So the last few minutes of the talk, I can probably do this in 12 minutes, talk about cytomegalovirus and glioblastoma. So, what is cytomegalovirus? 70% of the people in this room will have IgG that recognize this virus in their bloodstream. So the others probably haven't encountered the virus yet, but probably will. And when you encounter it, you will probably not have very severe symptoms for the most part. You might feel like a little cold or something. And the virus will then go latent, and it will stay with you for the rest of your life. And as it stays with you for the rest of things change a little bit. Your T cells change. They become skewed towards recognizing CMV. So as you age, and if you're CMV positive, 10% of your T cells or something like that will recognize CMV that are circulating because they're working to repress the virus. So and it can have devastating consequences in pregnant women or newly born babies that are infected with CMV. The CMV can enter the central nervous system, cause deafness. And it's a major public health problem. In HIV patients or in organ transplant recipients, CMV is a problem, as the doctors will know in the room. This is something you have to really watch for. And there are treatments for CMV. Valganciclovir is an antiviral drug which will help prevent CMV replication. Right. So there was an early study in 2002, which I haven't got the slide here, but they showed some staining of CMV, and that started a kind of ball rolling here. And what my lab did initially was stain glioblastoma specimens for CMV, and what we could see, if you use a standard clinical test, you can't see it. But if you use an optimized approach, it was slightly different fixation methods, all of this stuff. You can pick up a signal inside human glioblastoma. This is a positive control lung where you see it. Staining is a bit stronger, but in GBM you can see a light stain, and this is a immunofluorescence. And we got this interesting pattern. This is a CMV protein called PP65, and it forms these structures. We've done this. We haven't done this in enough patients yet. We've probably done this 4 or 5 specimens. That's all. But this is but this is consistent. Where we see this type of stain, and we thought this looks like a blood vessel, and so we used CD31, and what we saw was that it was close to a blood vessel, but not completely associated with the endothelium. So then we stained for NG2. NG2 stains a number of different cell types, but one of which is pericytes. And pericytes are smooth muscle type cells that coat blood vessels. And what we saw was this complete association of the CMV stain with pericytes. So that's interesting. We wanted to know if we could detect CMV by PCR. And nucleic acid sequencing, it's very challenging to detect this virus. It's probably at low levels and gets squashed out. So this is about as good as we've got with a very, very specific CMV PCR. Right? So normal brain is here, CMV is here. This is human brain tumor specimens. Very small amplification, but this is significant. These are some controls where we've looked at cell lines that are infected with CMV. This is the human CMV. And I'll tell you about our mouse model in 1 second. This is a mouse brain tumor in a CMV infected animal where we can detect CMV a bit more clearly, and in the mouse lung of the same animal we can't detect CMV. So we think that CMV somehow gets reactivated in a tumor microenvironment. That is a hypothesis. And so we set up a mouse model, and in the mouse model it's a standard GBM called GL261. We infect newly born mice with CMV intraperitoneally. It's a mouse CMV, not human. So every mammal has its own CMV, and they're all a bit different. So that's a potential confounding factor, but they're close enough. And what we saw was that these tumours, so we wait 16 weeks, so the virus disappears. There's a phase of acute infection, then the virus becomes latent, probably in the bone marrow, probably in other tissues too. And what we see every time, not every time, but the vast majority of the time, this is a very reproducible experiment because it's now been done by 4 different postdocs in my lab in different locations, so it really does happen. We see this significant decrease in survival in the context of CMV infection compared with controls, right? And we also, when we stain them, the most significant difference, so these are controls, stained with CD31 for the vasculature. These are 2 different models actually, CT2A and GL2C1. You can see very clearly there's a difference in the vasculature between the 2. These are very fine, more mature vessels, probably delivering blood much more efficiently to the tumor, which will allow faster growth. And this is an MRI technique that allows you to visualize blood flow in the tumors, and you can see there's a more intense staining in these tumors from the MCMV mice. Some more blood vessel stuff. So we found, at least in the GL261 model, that pericytes were coating the blood vessels. This is an indication of functionality of the blood vessels and maturity. So somehow, in the context of CMV, you're getting a more pronounced angiogenic effect, which is linked to tumor growth, we think. This is newer work, so we wanted to see if this was consistent across a number of different models. So these are all very distinct mouse tumor models. These have an intact immune system. They're immunocompetent models. And what we find is that in each case, we get the same effects. We get reduced survival with CMV and faster tumor growth. And again, very clear to see, So we did some mechanism. I'll tell you a quick bit about the mechanism. Yeah, we've got a couple of minutes. What we did, we took conditioned medium from tumor cells infected with CMV in vitro. These are human right now. And we put it on a tube formation assay. These are vasculature cells, and they form tubes in this assay system. And when you put the conditioned medium from a CMV infected cell on those, the tubes form much faster. So there's something in the conditioned medium that is driving that. And also pericytes migrate faster too with the conditioned medium. So there's something in that medium after CMV infection that is changing the behavior of these non-tumor cells. So we did some RNA sequencing just of infected tumor cells, and these are some of the changed genes. This is a list of ones which are known to be associated with blood vessels, right? So we just focused on those. And just as a decision, we looked at this molecule called platelet-derived growth factor delta, PDGFD, platelet-derived. This is a famous family of tumor-driving growth factors. We're not working on it right now, but I think we should, because we've got this really exciting data where we see that PDGFD is upregulated by Western blotting. This is after infection of cells with CMV. And we also see that we can drive, PDGFD alone can drive pericyte migration. And if you use immunodepleting antibodies, you block it. So it's got some role here. And then we did a CRISPR experiment. So we removed the PDGFD from our tumor cells. And what we found was the tumor cells, it wouldn't grow if you take PDGFD away. This is without CMV in the system. If you have CMV around, then you greatly decrease tumor growth, but they do start to grow eventually. So it's complicated, but this PDGFD appears to be an important mediator of some of the effects that we see. So we have a model. We think at least you could substitute mouse for human here if you like. So what we think is that the GBM, while it's growing, there's an immunosuppressive microenvironment where immune surveillance is no longer occurring. And if there's a cell in that tumor microenvironment that harbors the latent genome of CMV, then that can escape without immune regulation and can start to spread and can actually start to have effects on the biology of the tumor cells and the vasculature. That's our theory. And that we know there's some transcription factors involved that drive this. It's not only PDGFD, there's going to be other things. This then leads to angiogenesis, pericyte recruitment, increased blood flow. And then what we think happens is that there's a bit of a cycle where GBM grows more, there's more immunosuppression, there's more, and so on. And this might be a big driver of the aggressiveness of the tumors. I'll show you some more evidence for this. So we did some I won't talk really about this. This is just something we can maybe discuss later. We looked at the immune composition of these tumors. We see increased T cells here. We see increased dendritic cells and macrophages in the tumor microenvironment, the CMV-infected animals. This is through cytoff. And we also did some flow cytometry. We see increased CTLA-4 and PD-1. They're those checkpoint molecules that block T-cell activation, but also are involved in T-cell activation. So it's suggesting a more inflammatory microenvironment. We also see that 15% of the CMV, of the CD8 T cells in the tumor microenvironment actually specifically recognize CMV. You can do this using assays called tetramer assays that you might know about, where you put in a fluorescent molecule that shows what the target of those T cells is. And we know that 15% at least of those T cells in the tumors, in our models, recognize CMV. It's very interesting. And this is some other data. So this is from human patients with a group in the Netherlands that I work with. We got blood from them. They got some serum and they sent it to Boston. And we measured the CMV positive negative. There were 60% of Dutch people are positive for CMV. And then we split them into two groups, positive and negative. We saw here a change in progression-free survival. So CMV positive patients had a significant decrease in progression-free survival. And we also did gene expression analysis. The reason I put this in is a couple of interesting things is these genes all change in the mouse model and all change in humans too. So this is so overlap in the two functions. One of which is interesting is IDO1, which is an intracellular immunosuppressive molecule, which is considered a target in cancer too. So very interesting. This is still ongoing. We're trying to make a manuscript out of this now. This is something somebody already published, showing that CMV patients with IgG positivity, so these are, GBM patients with CMV IgG positivity. So so they split, like we just did, they split them into two groups. The patients with CMV do worse. Right? Not massively worse, but it's significant. We also showed in another experiment that we're trying to finish now, is that CMV drives upregulation of a protein called MGMT, which is involved in resistance to therapy in glioblastoma. And we could see that the CMV mice do not respond to temozolomide while the control mice do. So we've got some evidence that not only does CMV potentially drive tumor growth, but it may drive some resistance to therapy. And there's some clinical trials going on. So there's other people, I'm not the only person working on this. There's only a few of us, but there are clinical trials going on. There's a center at Duke University. There's Karolinska Institute. Karolinska is very famous. They're doing, I'm nearly finished. They're doing a study using valganciclovir alongside standard of care with primary glioblastoma. And what they showed was this very interesting curve. So the blue is historical controls. For those of you who are clinicians or those of you who look at this, historical controls don't mean too much because it's a kind of a bias. It's not really, you have to do the randomized trial. And the randomized trial has been going for 3 years, and so we should know soon whether this data is really true. So anyway, in their historical controls, they see a median survival similar to what I showed you earlier, 15 months. But if you give valganciclovir, an antiviral drug, alongside standard of care, you can get a 23-month or so median survival in these patients. So something is going on. Immunotherapy, this is an early stage immunotherapy trial using vaccines, showing a difference. However, this study has not panned out to my knowledge, so I don't think this is really working at this point, but may need some fine-tuning, right? And then just to wrap up, I'm not going to give you that. We did some more stuff in the mouse. We took an antiviral drug called cidofovir. This is not ganciclovir, it's cidofovir. It blocks viral replication. When we give cidofovir to the mice, look, we can increase the survival to the same level as the control. So what we think we're doing is we're reversing the effects of CMV on tumor growth. It's not curing anything, but it's reversing the effects of CMV. And so what we showed, we get the effects on the vasculature in the CMV mice. We reverse those with the drug. PDGFD expression decreases, and all of that pericyte coating decreases. So we think we're reversing the effects of the virus by administering an antiviral drug. And so what we're doing now, I'll just show you very quickly, and then this is the last part. Cidofovir is not a great drug. It's not very bioavailable. It doesn't get into cells. And so there's a company that has made a version of cidofovir which is modified. It's modified by adding a lipid chain to the cidofovir drug. The cidofovir is just a nucleotide basically, and it interferes with DNA replication. And so this lipid helps the drug get into cells and makes it much more bioavailable. And so this is actually a drug we think we can translate into patients. We're actually working with the company on this as my conflict of interest. I have some money from a company called SymBio in Japan who have helped support this, as well as the National Cancer Institute. And so this is more bioavailable. It has activity against many viruses, including CMV, and it's approved by the FDA to treat future smallpox infections. So it's like if there was ever a flare-up of an uncontrollable virus, brincidofovir may come into use for that. So it is known to be. And so what happened when we give brincidofovir to mice, actually, quite interesting, not exactly what I was hoping for. But here we see the untreated animals. This is a different model called SB28. The CMV infected ones do worse than the controls. And when we give cidofovir to these animals, in purple is the CMV and in red is the control. So actually this drug is increasing the survival of animals even in the absence of CMV. But we think there's probably more of an effect in the CMV animals. So this is something we're trying to address now using different dosing of the drug. And what we found is that cidofovir causes in cancer cells a massive amount of DNA damage. So it actually may have a dual effect. So on uninfected cells, it could be causing having its DNA damage roles, but in the context of CMV, it would also be reducing the effects of CMV. That's what we think. And so we're hoping that this might turn into a clinical trial in the near future, and I'm going to cut it off there and just tell you, we're looking at tumor initiation as well, but there's not much evidence. We've shown you that tumor progression may be a connection, and that further study of antivirals could be important. And there's my lab. And thank you to them, and thank you to all my funders, and thank you to you guys for listening and I hope you enjoyed that.
Thank you very much, Sean. That was wonderful. Is there any question here in the audience? Yeah, I think we have a question here. Victor? So Victor?
Thank you very much, Sean, for your comprehensive talk. I did enjoy it. We have a little team here working or interested in myeloid-derived suppressor cells. So I have two questions regarding your results. So one is whether, I read that some of the oncogenic mechanisms of viral infection is the recruitment or the amplification of these suppressor cells. So my question, or my hypothesis, would be that maybe CMV could also do this effect.
Yeah, so I showed you some macrophage. I think when we say macrophages in that experiment, it would include probably all of those as well. So we have some cytoff data that we're looking at, and probably we can pick that apart. I don't know yet, but I think that's a very good.
The other question is regarding the treatment. One of these the natural product, indigo. What's her name?
Indirubin.
Indirubin. Okay. Whether this could be another target of this drug, whether this drug could modify this recruitment of these suppressor cells or modify the function.
Yeah, that's a great question. So I didn't really. I couldn't show everything today, but in that paper we do some detailed studies, and again, we do see a reduction in the recruitment, and we see more T cells again. So we think that compound is favoring an anti-tumor microenvironment, and that could be the mechanism.
Just thinking about things that we can collaborate with.
Yeah, love to. We can share everything.
Sure. Thank you, sir.
Thank you, Victor. Is there any other questions in the audience here? Yes, Claudia.
Thank you so much. I learned a lot. So I was wondering about the quite a paradox between your two projects. One first is the need for opening the BBB barrier. So you need to open the barrier for the drugs to get into and do their tumor effect. But on the other hand, when you open the barrier, you are allowing all these reservoirs, immune reservoirs of T cells to get into the brain and unlock their transcription of the viruses they have integrated in the genome. So, and they are enticing that loop, endless loop of.
So you're asking if opening the blood-brain barrier would make things worse in terms of the immunological consequences? Yeah, that's a great question, of which do I know the answer? So we've done. I think that it probably depends on the method that you use to open the blood-brain barrier. So the biodrug that I told you about, which has these multiple effects, probably may have some, there may be some compensation in that bio might might suppress immune cells anyway, or it's anti-inflammatory, so that could counter those effects. But also, I am not sure how opening the blood-brain barrier really affects immune recruitment, because immune recruitment requires cytokines, it requires a lot of really complicated, it does require kind of a number of different steps, right? So if you open the blood-brain barrier, will you really lead to a big change in the tumor microenvironment? I don't know the answer to that, and so there is some literature that will provide an answer. So the focused ultrasound that I mentioned has been done in animals and humans, and I know there's some single cell data that I haven't really looked at yet, but that might give you an answer as to whether opening the blood-brain barrier would be dangerous. But you made one other good point I want to mention, is that what I think is happening in the CMB model is that things like myeloid-derived suppressor cells and monocytes are probably carrying the virus in as well, and they're probably the culprits. So we did one experiment where we took bone marrow from CMV-infected animals and we put it into CMV-naive animals with tumors, and then we were able to detect CMV within the tumor. So that tells you that bone marrow could be carrying the CMV into the tumor microenvironment. Yeah.
Question here, please.
Thank you for your presentation. Do you know if there's any evidence of some interaction with some vaccines that are effective against CMV? Just an example for example, the herpes zoster from shingles.
Yeah.
Is there any evidence that affects to cytomegalovirus and then to the brain tumor? And on the other side, if there's any vaccine that are used to cytomegalovirus in order to avoid those?
Yeah, so there's a few trials have been done with vaccine. The one was published recently in Nature Medicine from this Dew group, and they used dendritic cells pulsed with the CMV, one of the CMV proteins, the most immunogenic one, PP65, and they've used those as a vaccine. But the actual data doesn't support at this point that can reduce tumor growth. But I think it's something that probably needs to be worked on, and maybe you need not just a vaccine, but another combination, maybe because those tumors are very immunosuppressive. So is a vaccine going to be enough to allow the immune system to really do its job in the tumor? I don't know, so we're trying to do things to the tumor microenvironment. I think that might be how you get those to work. But the other thing, you mentioned chickenpox. There's a good study where it's a retrospective study, so it uses very old patients and to get the volume of samples needed. And they look at chickenpox, they look at herpes virus 1, 2, and CMV in patients and how that affects outcome. It's a guy called Steve Francis from UCSF. And they see so we think that CMV promotes tumor growth. It seems like varicella, it works the other way. So patients who have been infected with chickenpox have less glioblastoma. And that's one of the most significant findings in that paper. So it seems to be a real finding. And the people speculate maybe there's a chickenpox antigen that's somehow shared between chickenpox and glioblastoma, but that's really not known yet. But it's a great question, actually. Yeah.
We have a question here.
Thank you very much. I enjoyed the talk. I'm not a cancer guy, but I just have a question. Have you checked calcium channel activity in endothelium cells?
But you are a cardiovascular person, so
Yes.
yeah. I'm always scared when there's a vascular person in the audience because they probably know more about those cells than I. No, we haven't. We haven't actually looked at calcium channels. Do you think it's worth looking?
Yeah.
Yeah. All right.
I need to know.
Yeah. And I assume it technically it's probably quite straightforward. Yeah. Yeah. Good.
Nice
Thanks.
talk, Sean. I have a question about CD8. Did you check the functionality of CD8 in the same V model?
No. So functional. You mean taking them out and seeing if they make interferon gamma? No. It's another experiment. Yeah.
And did you check if there is any NK cells, natural killer?
There are NK cells in our panel, and it looks like they go down a little bit in the context of CMV, but we haven't really thought too hard about those. But the interaction of CMV and NK cells is really important. So some of the early work discovering NK cells was all related to CMV because they are a very important aspect of that. So there's so many questions. Yeah. Yeah.
And did you check the immunoevasion that they produce CMV? For example, this model, if they produce like this is already stage. For example, the tumor they grow because of the CMV they produce this they call immunoevasion.
Immunoevasion. So yeah, we see. I think what we see is. We see an attempt at an immune response to the virus, but I think it fails because of the immunosuppression in the microenvironment, and that's about as far as I can go now. But I think I would appreciate. We need to have more immunological insight into this process. I think it's a critical part of it. So we see things like granzymes and perforins elevated at the gene expression level. So that's telling you that something is happening, but it's not. It's not getting rid of the tumor in this point. Although so we've had some technical issues with one of the models. GL261 is considered very immunogenic for. I don't think the reasons are truly known, but sometimes that model fails to establish. It's like and it would be interesting to know why that would be. Yeah.
Thank you.
Any questions? Okay, I have some couple of questions, very quick ones. Yeah, I think it's very interesting. I can see a lot of overlapping with what we do also. Is there any reason why we see CMV with blood vessels? Is it because it's just the initiation of the infection? Because you said you have 16 weeks and then suddenly dropped. Do you think if you see patient samples, you may not see that particular features when you see it in vivo?
Yeah, I've been trying to work that out. So endothelial cells infect quite well in vitro, and they actually make the tubes faster when you just infect them. Okay. But the reservoir, and people have seen CMV infection of the endothelium, is known in the literature. But there are some species difference. So in the mouse, we see CMV in the endothelial cells themselves. In the human, we don't see it. We see it probably in the cells surrounding the vasculature. So there's a bit of a difference there.
Maybe there's a progress probably there.
Yeah.
And that probably the time course could answer the
question.
question.
And then I have some virologists who I work with who they don't understand CMV entirely by any means, but they think that it replicates very slowly in monocytes, but it can release itself, and then if it infects but it can spread very quickly in endothelium. So this is one of the potential reasons why you see it in the endothelium. It can spread quickly through the endothelium. But we don't really. There's many questions. There's enough to keep us busy for a long time here.
Yeah, no, I understand. I mean, just was curious about it.
No, it's good.
For the BTB barrier, so we know that the big challenge for neurosurgeon is basically the edema. So the blood surrounding the tumor, and I know the GL261 model creates huge edema around the tumor.
Yeah.
So how this basically with CMV, is there any particular reason how basically you can, because you have shown some downregulation of the time junction in BTB.
Yeah.
Have you looked to the edema
Yeah, it's an obvious question. So
Yeah.
what's the BTB like in CMB?
Yeah.
We haven't really, we need to, the reason I do many projects is so I can do those kind of experiments where you
Yeah,
take,
yeah.
but we actually haven't done that.
Okay.
It's a question, sometimes people who work in labs, if your boss says, can you do that? You should do it. So I sometimes ask my lab, do you think we should look at the BTB in the CMB model?
Yeah.
And then they just carry on with their own projects. So it is a really good question.
Yeah,
And the edema is probably less in that model, I
Yeah.
think.
So you have used quite a few drugs there. What is your best one?
My best one.
The best one that give us a hope.
So I'll tell you what the one that I think is really so. There's a some clinicians don't like it when I say it, but there's a drug that was tested in glioblastoma 10 or 15 years ago called dasatinib, and dasatinib is a small molecule that inhibits SRC kinase. And SRC kinase is upregulated in cancers, and it's FDA approved for breast cancer or some of the. So it's been in humans. It does the thing. When we put that drug in our BBB spheroids, we're able to see permeabilization of the spheroid, and the animal studies are kind of underway. I'd say we should know soon whether it actually does that in vivo. So it's a safe drug, and we could permeabilize the blood tumor barrier, and then we could potentially get a fast track. That would be, and
Okay.
that would be my favorite choice right now.
You mentioned the TP10 analog.
Yeah.
So I guess you're planning also to do some temozolomide.
Yeah,
on
so
that.
the TP10 is interesting. So that's the peptide that gets into the brain, right? So we've tried a number of things with that. So when you're doing drug conjugates, it turns out that depending on the drug that you add, you can change the properties of the peptide, right? So you put we've tried some other drugs, but then the peptide no longer has the ability to get across the barriers, probably because its structure has been changed by the presence of the drugs. So we have to be very, very careful about what we put on those, and that's a limitation, I think, of that approach.
Okay.
Yeah.
So in terms of the database that you're using, is there any particular database for patients that have been infected in the past?
Oh, that's not available
Okay.
to my knowledge in databases. So we have to do the same. We would use databases, for instance. So we haven't found CMB in any of the nucleic acid-based databases.
Okay.
It's not there.
Excellent.
I mean, but they're doing 25 million reads, not 100 million. So that could be a possibility. It's not going deep enough to find the virus. Yes. So yeah.
Excellent. Thank you very much. Is there any other question in the audience here? Or we can continue.
Great.
discussing.
Thanks for the lovely
Thank you very much.
questions. Great.
Thank you.
Okay. You're welcome. Okay. Very welcome.
Great.
Well, was I good? Hopefully it wasn't too long. I have a tendency to put too much. Oh yeah. You want this?